Today's physiology topic is apoptosis or programmed cell death. So this topic will be discussed under the following headings. First of all, we will see the introduction or definition of apoptosis and what are the various examples, that is both physiologically and pathologically seen in the human body, and the biochemical features involved in the process of apoptosis and the morphological features involved and the actual mechanism of apoptosis or programmed cell death. Apoptosis is defined as a pathway of cell death induced by tightly regulated suicidal program. That is, the process is started or induced within the cell itself and it is tightly regulated. And this word tosis, that is derived from Greek language, meaning shredding off or fall off or dropping off. So this apoptosis is a tightly regulated programmed cell death which is self-induced, and this is the active form of cell death which require energy, and this process is mediated by caspases, which are a family of proteins, and this process apoptosis does not elicit inflammation like the typical necrosis. Necrosis is cell death which is involved with inflammation of the surrounding cells. And this apoptosis acts at the individual cell level or small clusters of, or that is group of cells, and this apoptosis will be balancing the mitosis. Mitosis is cell division and apoptosis will be balancing, that is it will neutralize the increase in number of cells by decreasing the number of cells caused by apoptosis. So apoptosis is also called programmed cell death, which helps in deletion of unneeded or extra more number of cells which are produced during the embryogenesis. And it is involved in the normal involution process, that is shrinkage of cells or organs during the elderly age, and regression of hyperplasia or tumor. Hyperplasia is the increase in number of cells in a particular organ, and the tumor is also the same thing, whether it may be benign or malignant. There will be more than required amount of cell production will be seen, and the removal of viruses which are harmful to our body and in producing the immunity. So in all these situations, this apoptosis will be. So the key player in the entire process of apoptosis are the caspases. So these are a group of proteins, so which are derived from the cysteine dependent. C is taken from the cysteine dependent and aspartate directed, that is the first three letters ASP, and then proteases. So from these three words, the word caspases has been derived. So what it is? Cysteine dependent aspartate directed proteases. Okay, so remember the full form of caspases, which are the main role players in the apoptosis. So the examples of apoptosis, both we have to consider the physiological and pathological conditions which involve the physiological situations where apoptosis is seen are. So this is a physiologically means it is a normal phenomenon. So this normal phenomenon of apoptosis, it serves to eliminate the cells that are no longer needed. So this condition where it is seen in our human body. So first of all, if you take from the embryo to the production of a complete infant, that is embryogenesis, implantation and organogenesis. So in this entire process, the extra number of cells which are produced are deleted, or cells are being died by the process of apoptosis. And then hormone dependent involution, that is shredding of the menstrual endometrium during the menstrual cycle. And then cell deletion in proliferating pools, that is the cells will be continuously produced in the intestinal epithelium, and the excess cells, which are older cells, will be deleted or died off or shed off. and death of senile cells, that is older cells, elderly cells, and elimination of self-reactive lymphocytes, that is autoimmune, the lymphocytes which are produced which are reacting to the self cells. So those are also deleted by the apoptosis. And cell death induced by the cytotoxic T cells. So cytotoxic T cells are the type of immune cells, that is in the WBC there are various types of granulocytes, right? In those we have the lymphocytes, in those again we have the T cells. T cells are helper T cells, cytotoxics, various types are present. So the cytotoxic T cells will cause cell death, so that is also induced by the apoptosis. So these are all the physiological situations where the apoptosis is the main factor which helps in cell death of all the extra number of cells. So coming to the pathological situations where the apoptosis is seen. So pathology means abnormality, some disease or infection is present. So in which conditions the apoptosis is seen. So first of all, the most common is an injury or any accident, any abrasion, any cut by the knife or anything, any small break in the skin will lead to cell death of those the cells which are present in that injury area, injured part. And cell injury in some viral diseases like viral hepatitis. So those cells are also being apoptosed, or programmed cell death takes place in those infected cells. And pathogenic atrophy in some organs like duct obstructions. So ureter or any duct, if you take any duct present in the body, if there is any obstruction like in hepatic tract, so that will be blood supply or some supply will be lost to that organ. So that organ will be atrophied. That means because of loss of functioning, that organ will become malfunction or non-functioning. So there also apoptosis takes place, and cell death in some tumors. So now we will see the biochemical features involved in the apoptosis. These apoptotic cells usually exhibit a distinctive constellation of biochemical modifications that underlie the structural changes in the cells. Okay, so this apoptotic, that is cell death, cell death which is being programmed in a particular cell, they exhibit a constellation, that is a group of biochemical modifications that underlie the structural changes in the cells. So what are those structural changes? We will see protein cleavage takes place, activation of endonucleases take place, and phagocytic recognition takes place. So these are the structural changes which lead to the distinct major reactions or major changes, biochemical modifications will take place in the cell. The protein cleavage occurs via activation of several members of cysteine protease family, that is caspases, and it activates the several proteases which act on microfilaments like laminin, actin. So this laminin, actin you have seen in the cell membrane, and the breakup of nuclear scaffold and cytoskeleton will take place because of this activated proteases. And after protein cleavage, there is DNA breakdown, that is the DNA will be completely broken down. So the apoptotic cells exhibit the DNA breakdown of into 50 to 300 kb pieces, and the intranucleosomal cleavage occurs which into multiple fragments by calcium and magnesium dependent endonucleases. This resembles ladders on a gel on agar gel electrophoresis. So this electrophoresis is a biochemical mechanism. So you'll be learning about this electrophoresis mechanism in the biochemistry and also physiology. So when the protein is when this DNA is being placed on this gel electrophoresis, it resembles like ladder. So this phagocytic recognition also takes place in the apoptotic cells. How means apoptotic cells express the phosphatidylserine substance in the outer layer of the membrane, cell membrane. So this phosphatidylserine will be recognized by the macrophages. So this allows for early recognition of apoptotic cells, resulting in phagocytosis of this apoptotic cell by the macrophages without the release of pro-inflammatory cellular components. So this pro-inflammatory cellular components are normally released by the inflamed cells, but here there is no inflammation, right? So without the release of any inflammatory component, only with the help of phosphatidylserine produced in the outer layer of the apoptotic cell, the phagocytosis of these cells has been taking place. And next, the morphological features of the apoptotic cells. The cells will begin to shrink and round up, and they become more dense and detached from the neighbors, and the chromatin becomes very dense and separates into homogeneous masses, and cells will undergo budding and blebbing. That is, budding, like you have seen in your botany, budding of the plants, and blebbing is formation of blebs. Buds break off to become apoptotic bodies. Okay, so first they become shrinking and rounding off and becoming more dense, detaching from the neighbors, that is separating from the neighbor cells, and chromatin, which is present inside the cytoplasm, it becomes very dense and separates into homogeneous masses, and the cells will undergo budding, dividing off, and then blebbing. So these buds will break off to become apoptotic bodies. And phagocytosis occurs by the help of macrophages, that is the surrounding cells. And removal of apoptotic bodies takes place. So you can see cells round up and nucleus collapses. Uh, the chromatin condenses and DNA will be breaking down or fragments. Plasma membrane blebs, blisters and blebs form. So these are the blebs, so, which will be broken down, separating off and forming the apoptotic bodies. Okay, so this will be engulfed by the macrophage. So this also again same, the nucleus condensing, bleb formation, nucleus fragmenting, and the apoptotic body separated and being phagocytosed by the macrophages. Coming to the molecular mechanism of apoptosis. So the initiation phase is divided into intrinsic pathway and extrinsic pathway. Extrinsic is mediated by TNF, that is human necrosis factor, and FAS, and both these will lead to caspase activation, and the execution phase takes place, that is clearing of the dead cells. So in the both extrinsic and intrinsic pathway, we will see what happens. So there will be death ligand, death receptor will be present, adapters will present, disc formation will be seen, and the caspase 8 activation will take place in the extrinsic pathway, and in the intrinsic pathway mitochondrial changes will take place. Apoptosome will form and caspase 9 will be activating, and then 3 will be activating. And in one more pathway, that is perforin or granzyme pathway, where the perforins, that is through the cell these pores will be formed. So these channels are called perforins. So granzyme B and A will be activated, which lead to caspase 10 activation, and A will cause the DNA cleavage. So all this finally will lead to endonuclease activation, leading to DNA fragmentation, and the nuclear cytoplasmal proteins will be degraded, and finally cytomorphological changes and formation of apoptotic bodies will be seen. So remember, main thing is the caspases which are present here. In the extrinsic pathway there will be 8 activation, intrinsic 9 will be activated, and perforin pathway 10 will be activated. So in detail we will see the pathways. Extrinsic or death receptor pathway initiated by engagement of cell surface death receptors. The type 1 tumor necrosis factor receptor is present, and FA is also called as CD, cluster definition 95. So this is the death receptors, and this FAS is cross-linked by ligand, that is FASL, and 3 or more FASL molecules come together and their cytoplasmic death domains form a binding site for FADD. So this FADD then binds to procaspase 8 or 10 in humans, which cleave each other to become active. And there will be initiation of caspase cascade, that is one after the other, one protein after the other will be activated, finally leading to protein cleavage, cytoskeleton damage, DNA fragmentation. All those will be the subsequent situations. And in the intrinsic, that is mitochondrial pathway, what happens, there will be withdrawal of growth factors or cell stress will be present, and there will be increased permeability of mitochondrial membrane, which result in increased permeability and release of pro-apoptotic molecules into the cytoplasm. Leakage of cytochrome C takes place, and which binds to IPF1 in the cytoplasm, and caspase 9 is activated and the cascade takes place, leading to apoptosis. So in the extrinsic pathway, there is the death receptor, tumor necrosis factor and phase receptors are present, and the ligands will be seen. And in the intrinsic pathway, the mitochondrial membrane will be permeability will be increased. And cytotoxic T cell or bypass method. So this bypass mechanism for cells that refuse suicide via extrinsic or intrinsic. So the cells that refuse via both the extrinsic and intrinsic pathways, they undergo apoptosis by this cytotoxic T cell method. So here the T cells recognize foreign antigen present on the infected cell membrane. On recognition, this cytotoxic T lymphocytes release perforins. So these perforins are the cell channels which perforate through it, that is cell membrane. So the entry of enzyme granzyme B, a serine protease, will be allowed. So this granzyme B cleaves proteins at aspartate residue and activates several caspases. So this is most commonly seen with viral infections. So remember, this cytotoxic T cell method is most commonly seen with viral infections. So this is again one more picture for extrinsic death receptor pathway. You can see FAS receptor, FASL is ligand, and TNF receptor, and these are the ligands, and FADD activating procaspase 8, and here also 8, and this TRAF will lead to this pathway activating the caspases 8 and 9. Okay, and this is intrinsic pathway where the intracellular stress is seen. Mitochondrial membrane will become more permeable, leading to release of cytochrome C, activating the procaspase 9. And again, the apoptosis will be finally taking place through both these mechanisms. And this is a histopathological picture showing the apoptotic bodies. Okay, you can see the arrows are shown. And finally, don't confuse the apoptosis with necrosis, where the necrosis is the death of the cells and tissues while body is whole, still living. Note, this is an inflammatory response. Some cells and tissues are dead. So necrosis, there will be inflammation, remember. And autolysis is also called self-digestion, destruction of tissues by certain substances such as enzymes that are produced within the organism. So this is autolysis, which is self-digestion. And necrosis is death of the cells and tissues while the whole body is still living. So the differences between the apoptosis and necrosis. So apoptosis is very neat. Nothing, any chemicals or substances are not released by the cells. Simply the apoptotic bodies will form, which are engulfed by the macrophages. And necrosis, lot of inflammation, pus and everything will be seen. The chemicals are released from the cells. So this is very haphazard, ugly looking. Okay, so both are different. So that's all for today. I hope you understood the topic apoptosis. So read this topic from the standard books, that is Guyton and Genong, and then your reference books Byrne and Levy, Best and Tyler. Okay. Thank you everyone.